Good afternoon, everyone. We would like to start our first panel. We will be discussing the hubs for the development of and production of mRNA vaccines and other therapeutic targets. I have three esteemed colleagues and friends with me. Patricia Neve, who's a scientific leader for mRNA vaccine, Synergium, who is a technology transfer manager at Synergium, and Karen Fenner, Dr. Karen Fenner, I should also mention they're all PhDs, who is the executive director of the mRNA vaccine hub at Efrigen. Just a little bit of household management. Questions need to come through either the chat on the app, or you can raise your hand and one of the assistants will take your question. And I'm not going to be rude looking at my phone. I'm just going to look for questions coming in. So I'm with you. I'm not checking my texts. So without further ado, we'll go with Karen.
Good afternoon, everybody. I'd like to say a very big thank you to the symposium organizers for the invitation, for Efrigen to participate, and also a very big greeting to the VIPs, the directors of Fiocruz and Biomanguinos, and to Soterius and Patricia for all the organization and to your team who helped us. So as Rami said, my name is Karen Fenner. I am from Efrigen Biologics. We are a relatively small company based in Cape Town in South Africa. And due to the short time that we have, I've only prepared a few overview slides just to give you a brief introduction to Efrigen and the activities that are surrounding the mRNA hub, which is hosted by Efrigen. Right. So as I said earlier, Efrigen is a biotechnology company. We've been in existence for about 6 years. We have a shareholding in Efrigen, which is by Avocare Health Group. They own 65% share of Efrigen, and the IDC, which is the Developmental Finance Institute of South Africa. I cannot see the slides. Maybe I can move over. Right. So when we talk about Efrigen, often we say that it's a first for Africa, and this is because we established the first formulation center in Africa. Primarily, it was focused on adjuvant formulations, which are an essential component of vaccines, used to deliver the vaccine or to modulate the immune response. And further building on that platform, we have developed formulation technologies for complementary and alternative medicines, vitamins, minerals, and other healthcare products. And so in Efrigen exists two main platforms, which include this formulation technology platform, as well as the mRNA platform. And our strategy is to vertically integrate all the way from R&D to forward fill finish and to commercialization. And so to answer the question around what was the significance of Efrigen being selected to host the mRNA hub. So actually, how do you go back? What I wanted to say in the very beginning is that the mRNA hub hosted in Efrigen was born out of the initiative in COVAX of the working stream 3, which is around vaccine manufacturing. And it is a public-private partnership consortium, which mandate is to advance mRNA vaccine development and manufacturing for low-income countries. And the partners that are involved include the Department of Science and Innovation of South Africa, as well as the African Union and Africa CDC, Efrigen, Biotech, which is the spoke, which is based in South Africa, also in Cape Town, the Medicines Patent Pool, the World Health Organization. And so the World Health Organization and the Medicines Patent Pool really leading the initiative, and then the South African Medical Research Council. And so back to the question around why this consortium, and particularly Efrigen being the host of the hub. Really, there were a number of reasons, and like Bia Manguinos and Fia Cruz, who went through quite a rigorous technical due diligence by PDVAC, which is an independent advisory committee to the WHO. The additional criteria that we believe contributed to the success of the hub was initially our motivational foundation, which was really about looking forward to or establishing local manufacturing for vaccines in South Africa as well as on the African continent, due to the increase of infectious diseases and zoonotic diseases, and also the lack of adjuvant formulations. This is one of the reasons why Efrigen was established. And we believe that through local and regional manufacturing that you could increase security of supply, quality and affordability of vaccines and adjuvants. We also had two shareholders which really had a mandate for development. So Avocare Health and the IDC, we have a strong focus for development within Africa, and that was a strong support for the selection of Efrigen as the hub. And initially, as the company was being established, we had a very outward-looking collaborative culture in the company, and had established a number of collaborations locally and internationally. And then at the time of the expression of interest for the hub and the spokes, Efrigen actually had a vaccine manufacturing facility which was ready-made and available to receive technology. So at that stage, we had constructed a vaccine facility to advance our own vaccine pipeline. And with the support of our shareholders, we're able to redirect that facility for the purpose of the mRNA hub. So just to give you an overview of the South African consortium, the main objective is to work towards the submission of market authorization application for the COVID-19 mRNA vaccine, and to enable technology transfer to all the spokes. We have our first goal is to prepare a target candidate, a first generation candidate, which is based on a forward innovation of the Moderna vaccine, mRNA-1273, and simultaneously to develop a second generation candidate, which seeks to address lowering the cost of goods to improve the stability of the mRNA vaccines, and to enable freedom to operate in the hub as well as all the spokes in their low middle income countries. So let me see if I can get this to work. Okay, so what we've got on the slide is basically a summary of the three implementation partners within the consortium, being Efrigen hosting the hub, Biotech as the South African spoke, and the South African Medical Research Council. And this is a high-level overview of the sort of process that we are following. So in Efrigen, to develop the technology at lab scale, and then to implement this process development manufacturing, to do the process scale up, and then to prepare clinical trial material for phase 1 and 2. And with that, develop a technology transfer package that can then be disseminated to the various spokes. And after that, there is a time here where we refer to as optional scale up. So if you think about the various spokes in different regions, I'll show you a slide on that a bit later. There would obviously be different market sizes and markets that would be addressed by the various spokes. And so scale up would need to happen to accommodate that. And simultaneously to doing all of that, develop the analytical methods, establish the methods, and then do the characterization. And over here where we've got Biovac, you can also sort of instill all the spokes, because this would be the same sort of strategy. So to ensure that we have operationalization at the spokes, manufacturing process validation, and then each of the spokes would be the sponsors for the phase 3 clinical trial material going on to the phase 3 study and then market authorization. At the same time, I spoke about the second generation of vaccines. And so the South African Medical Research Council will coordinate this long-term research and development program, which seeks to address, as I said, bettering of the current mRNA COVID vaccine, but also looking at what are the next priority disease targets. So we heard some of the other panel members talking earlier about what happens after the pandemic. And so this is what this program seeks to address, is to make sure that the full value chain of vaccine development is seen, and that we can look at what are the appropriate targets, particularly for the various spokes in the regions that exist. So this is a very brief summary of all the activities that are currently happening at the hub, just all the work packages, which are components of the integrated product development plan. So we have process development, the preclinical program, which includes immunogenicity studies in mice, and safety through the GLP toxicity studies, which will be happening in rats. And then, although the challenge studies are not considered to be on the critical path for phase 1, we do believe that it helps to build confidence in the product. And so we would be developing that as well. And then chemistry, manufacturing, control, and analytics, which includes everything from equipment to scale up, particularly the LNP formulation, all the way through to full finish. And obviously a big aspect of this is the quality and regulatory aspects of this. And I believe that we will have a session a little bit later, which will cover some of this. But within Efrigen, we have to look at facility readiness and all the other aspects that go with it, and an opportunity to optimize and refine our quality system to also incorporate the mRNA vaccine manufacturing. And then lastly, the aspect of training and technology transfer, which is also an essential objective for the hub. So there are now 14 spokes that have been announced through the WHO. The last one, which was announced, is India. And the spokes that have the orange star are the ones that we have received in our facility for the very initial training and startup support, which has been coordinated through the Medicines Patent Pool and with Efrigen. We received Argentina, Synergium, and Biomanguinos in the Efrigen facility in the beginning of March. And then we had Indonesia, which is Biopharma, were with us in the beginning of April. And we will have Bangladesh, which is in the middle of May. So many activities all happening concurrently. And then this is my last slide, which basically summarizes the strategic alliances and cooperative research and development agreements that we are engaging in. Not really at liberty to say who these partners are, but just to summarize the various aspects that we are looking to address with these partners. These include process improvement and fast tracking of GMP production of the first generation COVID, sorry for the typo, 19 vaccine candidate. Also looking at process optimization to reduce cost of goods and improve thermostability, novel immunogenicity targets. So here we're looking at, well, what kind of priority disease targets are tractable with the mRNA vaccine platform? And so to say, these are kind of looking at the low-hanging fruits, looking at freedom to operate with various lipids. As you know, many of these are proprietary. And then also for the various spokes to look at how we can use the benefits of these collaborations and alliances to contribute towards other vaccine candidates that would be developed through the mRNA platform, and then a long-term development for improved processes and disease targets. And so I'd just like to end there and say thank you very much for your attention and look forward to interesting questions.
Thank you, Karen. Next, we have Herman from Synergium.
So good afternoon, everyone. My name is Germán Sánchez Alberti. As Rami mentioned before, I work in Synergium Biotech as a, well, I have just changed my job position to an innovation and development manager. My previous position was MTech manager. First of all, I would like to thank Fiocruz and Biomanguinhos for the invitation to be here in this meeting. So my talk will be about mRNA vaccines, and the subtitle is From Conceptual Design to Practical Result, because from the very beginning we did it in the conceptual way, and now we're having the first results. So, well, the topics that we're going to see during this presentation are first an introduction of Synergium Biotech and Map Science. Then I'm going to comment the first step that we perform and how we prepare for this project, and finally some results. Basically, this project started on 2021 on September, when Synergium was selected to become one of the spokes. For this project, we associated with Map Science, which is a sister company that shares some of Synergium shareholders. So I'm going to introduce both companies first. We did have experience on vaccines and monoclonal antibodies, but we were quite new to the mRNA technology. So we started. This is the reason because we started with a conceptual design and trying to understand first the basic science of the RNA technology. Fortunately, then we could travel to South Africa and visit Efrigen in March, and after that we could perform some results. So who we are, Synergium Biotech and Map Science are specialized in research, development, and manufacture of biotechnological products, biopharmaceutical products. Map Science was founded in 2008, develops and produces biosimilars using the latest innovation in upstream and downstream. Also, they produce the drug substance for AstraZeneca vaccine during the COVID pandemic. And Synergium Biotech was founded in 2010 after the flu pandemic. So the main core of our facility is influenza vaccines. And currently we are supplying influenza vaccine to the Ministry of Health of Argentina and perform fill and finish operations for several other vaccines in collaboration with companies such as Pfizer, MSD, and Seqirus. Well, this is a nice picture of the hub. We have Synergium in the middle, and Map Science is pretty close to our facility. Let's see some numbers. We have produced more than 170 million doses and more than 700 batches manufactured with 0 rejected batches. This is quite important for us. We have a balanced staff between approximately 40% are women, more than 300 direct employees. We have performed 5 agreements with multinational pharma companies, 8 successful vaccines tech transfers, and we have 6 public and private research and development projects ongoing. Map Science, they produce 180 million doses of drug substance for the COVID vaccines during the pandemic. They have 3 different drug substance manufacturing facilities. One is in Spain, and 2 are in Argentina. They have 1 European Union registered product, more than 25 approvals, country approvals, and 12 health authorization GMP certification Health Authority GMP certification, sorry. They have performed 6 tech transfer, successful tech transfers. So I'm going to run through this, just some of these numbers. Our main core is trivalent influenza vaccine for the Ministry of Health for Argentina that we produce in a partnership with Seqirus. But we also produce pneumococcal vaccines as a CMO for Pfizer, the Prevnar 13. We have produced 31 million doses from 2012 to last year. We have also produced HPV vaccines. Besides the Ministry of Health, we have also present ourselves to the revolving fund of the PAHO, and we supply 2 million doses of influenza vaccine during last year and 2.2 million of doses during this year. I'm going to show you some slides for our facilities just to see. This is the upstream size of mAbxience. Our plan is to produce the mRNA drug substance in mAbxience and the drug product in Synergium Biotech. So these are nice picture of our sister company. The downstream side also, it's a well-equipped facility. Both has cutting-edge technology and single-use technology. Synergium has 2 different filling lines. One is for prefills and the other is for vials, and a future project that is going to be ended for by a fully automated vial line by the first quarter of 2024. Well, the quality control of both companies are related to control vaccines and monoclonal antibodies. We have 2 different research and development teams with 50% are PhD and Master of Science degrees. So the first step was to define the team for the mRNA project. In this, we not only associate with mAbxience, but also we have some collaboration with the University of Buenos Aires, where they have a good basic research on RNA technology. And the first training that we performed was indeed in Buenos Aires University. Then we define a high-level project plan. Right now we are in the microliter scale, and we are waiting for some equipment arrivals. This is a nice picture during our visit of Efrigen in March. This is a summary of our activity. As mentioned before, we start with the training of Buenos Aires University, then we perform some process flow diagram, mass balance, raw material, equipment list, QC research, and finally, after the Efrigen visit, we start our research grade microliter scale. This is our basic process flow diagram for pDNA. We have some basic analytics in place also. Well, the first result that we have during the production of the pDNA. Here are some mapping restriction results and the IVT process flow that we are working right now for our research grade. Here we have some results for the IVT that we perform on a research grade. And finally, I would like to point out that we were able to replicate the mRNA production, at least at a microliter scale, and this was performed after the training in Efrigen. We are planning to move to a milliliter scale, and we believe that the next step should be related to roadmap definition for the hub and definition of the tech transfer package. We are waiting for the equipment arrivals to finish our microliter scale, and we are thinking about IVT optimization experiments. So this is all for myself. Thank you very much.
Thank you, Herman, very much. And last but not least, our gracious host, Patricia Neve.
Good afternoon. My name is Patricia Neves. I'm very proud to be here today representing Biomanguinhos in this initiative. And I would like to thank the organizers for inviting me to perform this talk today. And I will talk a little bit about our role in this hub implementation. First of all, I would like to say that Biomanguinhos is part of our mother institution, which is Fiocruz. Oswaldo Cruz Foundation has 121 years promoting public health in Brazil. So Biomanguinhos is one of the units of Fiocruz, which is much more than Biomanguinhos, but we are dedicated to immunobiologicals. So we are Latin American Excellence Institute in immunobiologicals, vaccines and biopharmaceuticals and diagnosis. In fact, tomorrow we will complete 46 years dedicated to the Ministry of Health of Brazil and dedicated to bring products to our public health system. Inside our development department at Biomanguinhos, we are able to go from applied research, performing all the way necessary to get the products to the market. It's a very unusual feature of the institutions. So inside Biomanguinhos, we have people from development, from quality control and quality management, and also manufacturing. And that's why we are committed, and it's possible to bring the product from the bench to the market. In fact, we are committed also to the world. Some of our products are pre-qualified by WHO, and we are able to supply more than 70 countries with our vaccines. And because of our skills, we joined this initiative from WHO to being part of this initiative for hub implementation. And then we were chosen by PAHO together with our partners in Latin America from Synergia and Mab Science, and together with Efrigen. So it's a picture of our visit at Efrigen on March, our team together with German inside the lab. So before joining this initiative, the Biomanguinhos development team were already compromised to start some work with RNA. In fact, we were working with cancer products. And as you may know, RNA products could be divided in two different types. The conventional one, that's the project that Efrigen is working on, and the self-amplifying one, where the RNA is delivered to the cell and it enters cycles of self-amplifying and amplifying also the antigen we are looking for. We are the antigen that is inside the vaccine and eliciting the desired immune response. Biomanguinhos was working on a platform for self-amplifying RNA, and we have chosen this platform because it has some advantages, which are enhanced and prolonged antigen expression, lower effective RNA doses, intrinsic adjuvant effect, but we have enhanced cross-presentation because of mechanisms of self-amplifying. And the most important feature for us to choose this platform was that this platform is an option for a single vector delivering multiple or complex antigens. As you can see here, in this construction, we could put in line more than one antigen of interest. That's exactly what we are doing for SARS-CoV-2. So Biomanguinhos has a proprietary plasmid and formulation designed here. We have plug and play. This plasmid is a plug and play plasmid, so we could take off and put on whatever we want, making this platform very flexible to other vaccines. And for SARS-CoV-2, we are working with 3 different constructions engineered to direct SARS-CoV-2 antigens to the desired cellular compartment. So as I said, we are working with 2 different antigens for SARS-CoV-2. One of these is the S1 portion of spike, aiming to elicit neutralizing antibodies, and the other one is nucleocapsid protein to elicit cellular responses. So with these constructions, we hope to overcome the problem of the appearance of new variants. This platform has also some advantages in terms of manufacturing, which are the low costs for operations and investment to start, and also low costs for the batches and low size of batches. Once the doses are usually lower than conventional mRNA, we have much more economy in producing this kind of vaccine. It's just a grammar to show you that RNA production is much more, let's say, simple than working with cell-based systems. It's a whole enzymatic process, and that's why I think it's the magic of working with RNA, because the nature of RNA never exchanges. So you could exchange the product, exchange the antigens, and the process to produce it will be the same. So it's the strongest feature of this platform we are citing here and with our partners. So here in Biomanguinhos, we are planning to use our pilot plant area to set up the RNA production area. So we have about 500 square meters with capacity of produce 100 liters of IVT per reaction. 1.5 lots of IVT is enough to produce 120 million doses of COVID vaccine, which is our actual demand for Brazilian Ministry of Health for COVID vaccines. And we will take 35 days to produce the lots. So it's a lot of economy and readiness and quick process. We are also taking advantage of our quality management team capabilities to take into account in this project all of risk assessments and QBD design to get to the people as soon as possible, having an accelerated regulatory approval. We are working with a very aggressive timeline. We hope to get to the phase 1 studies still this year, late this year, and we are setting all these things together. So we are going fast with the line of the project, and we are also setting up quality management, quality control capabilities, and manufacturing area. I'm just to let why we are interested in this platform.
Por que estamos interessados nessa plataforma?
But RNA market is increasing from 9 to more than 15 billion until 226. And I think that this initiative for hubs and this partnership between South and South countries is a big, huge opportunity to enter this technology and stay together to offer these products based on RNA technology to people who need. So here at Biomanguinhos, we are setting up all these skills inside all areas, R&D, QC, regulatory, clinical trials, and production to dominate this platform in Brazil. And together with our partners, we will offer this technology. It will turn this technology accessible. So I would like to thank our partners, Efrigen and Synergium Bioscience, to be here. All the team from Biomanguinhos who are supporting a lot this project to go fast. The technological development team, all labs and people involved, our supporters, PATH, MPP, and PAHO WHO. And of course, our wonderful team from Technological Immunology Lab. Here you have Thiago. Thiago is not more with us. I'm finishing, just a minute. He's at Porto, he got a position there, but he was the responsible for RNA design. And some of other people joined our team, but I would like to thank this great team to be with us in this journey. Oh, that's all. Thank you so much for your attention.
Thank you, Patricia. I have to admit that I feel very lucky working with a group of people dedicated to public health. So we have one question from the audience and one question from me if we have time. And I hope I spell the person's name right, from Sebastian Machado Weber. What's next after COVID-19 from a vaccine candidate? And any thoughts on the next targets with regards to raw materials and unique reagents and supplies? Pretty much 1 minute each. So, Karen?
Say that again? No, joke. So the next target. So we are working with partners to look at, as I said earlier, what are specific priorities in different areas. Obviously, for Efrigen being in South Africa, we would be looking at the heavy burden of disease with TB and HIV, but we acknowledge that those are very difficult targets, and that will be a longer term program. So you have to, apart from it being tractable with the mRNA vaccine technology, you also have to have the correlates of immunity. And we know that even with HIV, that's still very difficult. So, but then in terms of the other spokes in different regions, we are suggesting to look at what are the low-hanging fruits and what are the priority diseases. For an example, we heard earlier from Dr. Akuru about measles, for instance, being in Brazil, and we know that through some of the assessments that we've done, measles would be a very good attractive candidate to look at. And for South America, probably dengue and Zika. So that's just to give you an idea. And probably for the different spokes, they would look at what are the priority diseases in their particular area.
Yes, as Karen mentioned before, I believe that respiratory virus could be a target, of course. There should be some concerns in the region about, for example, Chagas disease. The good things about mRNA technology, I believe, is not only that you could deliver prophylactic vaccines, but also therapeutic vaccines. And this is something that we could explore for Chagas disease or also in leishmaniasis. And this is very interesting to explore. We are quite new to the technology, so I believe that this is something very challenging for the region. Yellow fever also, and well, there is a problem, of course, on raw materials acquisition because of the big companies have a big agreement with the suppliers. So this is something that we have to deal in this region, but well, we are in this way, I think.
I think that German mentioned something very important, that, as I said, RNA platform is very flexible. It's not only about preventive vaccines, but also therapeutic vaccines and maybe protein expression inside the body. We are seeing that the first works coming from academies saying that it's possible. So we have a large avenue in front of us. And that's why I think that this partnership is so powerful for low and middle income countries, because it's a game that we could win with people who need. Thinking about other vaccines, I think that the easy vaccines, the easy diseases they are already controlled. Let's say this, there are already vaccines for the easy disease. So the challenge in front of us are dealing with the difficult disease, like HIV, TB, and other, and even the disease who are neglected. That's our role in this game. So I think, and for raw materials, we are having a lot of problems to get. Then maybe if everybody is coming inside the RNA platform, it will be very important to have good suppliers to support these developments and production of RNA.
Thank you all. I just want to add one thing, is that WHO and Medicines Patent Pool are investing a lot of time and resources in securing sustainable supply of reagents and critical raw materials. And so the idea is not only to develop technology, but also help these manufacturers access these critical reagents. Because I think, as Herman said, right now, the large multinationals, from obvious reasons, basically are almost controlling all the critical raw materials right now. We have time for one more question. Okay, so actually it's my question. It's again 1 minute each. Your thoughts about the benefits of working together, or your wish list for partnering together. Kind of what do you see the strengths of working together and getting from this collaboration?
So I think that there was always an intention to have these South-South collaborations, and there was nothing like the COVID pandemic to actually sort of catapult us to making it happen. And I think that the mRNA hub and spoke is, although this kind of strategy has been used in the past, I think that the collaborative nature and the networks within the hub and spoke model is really unique. And one of the advantages is that with having the hub, if you can conceptualize it as a tube to channel technology transfer, not only from the hub to the spoke, but also from various spokes to other spokes. what it enables us to do is to really have a rapid response. So if you think in the case of the pandemic, where you have all these spokes, we can really join together to be a really strong force. And even in the cases of raw material supply and so forth, having a network means that we can rely on each other. And I think that is a unique opportunity for us to really take this forward, not only for pandemics, but also for other priority diseases.
Okay, so I found the concept of hub very interesting in terms of collaborations, because every spoke goes to the hub and train themselves in RNA technology, and then could go to the region and develop what we have trained previously. So I believe that this model is very, very beneficial for these regions. Of course, this was triggered about the pandemic. So the hub initiative in that case, I think it's a very good strategy to have a better response for the next pandemic in terms of RNA technology for the region.
I think that the most powerful thing that we have from this collaboration is that we are building capacity in South world to respond to other pandemics, and mainly we are learning together. That's what I feel. Because in Brazil, we say that we are exchanging the wheel while the car is running. And that's what exactly we are doing. RNA platform is very new for us, but we are not alone. The concept of the hub makes us learn together.
Thank you very much. Unfortunately, we are out of time. I know that there are more questions coming from the audience, so I urge you to reach out to these great people. The next session is on TCID50 as a validated tool to measure antiviral activity in functionalized textiles, which I want to listen to. So thank you very much.